I'm representing Paulo. I'm also a member of CTN Bio, as Luis Sergio and Maria Lúcia. I'm going to go a little bit deep on the regulation of gene-edited animals here in Brazil, since Maria Lúcia gave a more general overview of the structure of Brazil. However, the title of my Paulo's talk, which is mine now, was Familiarity. What can we learn from what we already know? And I have to confess that I was not familiar with this term. Well, I don't want you to be lose attention on my talk, and I really want to open the minutes we'll have later for discussion. So if you want to complement or say something, don't take my words as final words. It's just an attempt to put a context here in this session. So, yes. So first of all, we are here, I think all of us are scientists, at least in the beginning. Then some became more regulators, but we are scientists. And the science has a sort of its own evolution. Scientific innovations are daily incorporated in the technological repertoire. So what is science today, tomorrow is technology. It's sort of easy since the world is started. The regulation follows scientific progress and not come before. So we need to have the innovation, then we have regulation. It's impossible to de-invent something. We never heard about that. We invent and that's invented. Some things we block, like cloning human or whatever, different bad things that humanity face. We have unlimited innovative capacity, and science unlimited innovative capacity is the main feature of science. And we are talking about gene editing here, pretty much. Is something very hot topic. And 2020 chemistry Nobel Prize was on CRISPR. So this sets the scene and put us in a very clear context. So I confess, I try to learn more about familiarity. And I found this interesting document from 2017 from the National Academies of Science. Basically what they joined different subgroups of the National Academy, and they went through the what is necessary or what is going on regarding future products from biotechnology. And I found some slides that I want to put here just to say that I did I'm familiar with the familiarity. So first of all, we are here in terms of product complexity and novelty. So we are either doing GMO or gene edited with domesticated organisms, cow, soybean, corn. We know these plants and animals for several millennia. We are doing transgenic recombinant DNA, only few gene pathways, something very specific, and we can compare with the wild type and different comparisons. Then as we move to the complexity and novelty, we go to some undomesticated organisms. We can do new genome engineering. We can assess simultaneously multiple pathways. I was faced with one problem with microbes, that the company was doing modifications to make fatty acids. So they could do 1,000 modifications in 1 week, and they will have 1,000 new products to test. How they're going to submit that in a evaluation procedure to test the 1,000 microbes? So we had to, and we did in the CTN Bio, we made a normatization to deal with that. So they could do the research and come up with from 1,010, then from 10 2, and these 2 they can do more regulation. So these are new things. And of course, sometimes we cannot compare with the previous brother, sister, cousin. And then it becomes more complicated. Many candidate organisms, more different technologies like gene drives. We can do de novo genomes, or we can do cells synthesis without cells. Then we can have basically no comparison. And finally, we can have microbes totally synthetic. We can have plant and animals also with lots of changes. We have metagenome, microbiome, etc. And we have a totally different thing. So how we as regulators or scientists will deal with this? So it's quite complex. And all this was due to this famous graph here. This is the cost of DNA synthesis, double strand, single strand, and DNA sequencing. So basically technologies we have to use for our science work. This went down, this is a tenfold, right? Sequencing was very crazy, eh? Went very down. So with this information, we could do much more genomes. I remember when we and other colleagues in this room were in this meeting in 2002 or 2004, where we decided which genome we were going to do from animals, and it was cattle. Okay, let's do that because this is going to be the only one. Let's do it well. So we did cattle. Now we have a 1,000 genomes cattle project. We have genomes from everywhere because of this feature, this trend of science. And then familiarity. I learned, please correct me later, that familiarity has been a sort of a sense used in the US system for evaluation, where you have this entry point. I don't know if it's still valid or it has been changing, but you have to assess the US system using one of the concepts is the familiarity. Is this organism, this change is familiar or non-complex, or is unfamiliar and complex? So this is a system that is very specific. And I'm not going to tell about that because I can't. So I decide to use my minutes here to show something, but I'm not going to show again because Maria Lucia said how Brazil went from zero regulation to total regulation in a way that things move. And this is very important. When we come with numbers, Brazil is going very well with agriculture. This is due to several factors, but one is that we use technology as soon as it comes. It's we are very good and early adopters of technology. And I have to say the government has been, the system has been pushing for that. So this is how, was shown by Maria Lucia. It started in 1995. We are celebrating 25 years of CTN Bio. CTN Bio is not a thing. It's not even an institution. It's a group of people. It's more a thinking thing than a organization. There is no building. Few employees, fixed employees. Some of them will be here today or tomorrow. The rest are members who are volunteers, are selected, picked, and they do the work for zero money, only based on knowledge. So it starts in 95, and there were, as she said, 3 moments. The first, the setting the scene, setting the first law, which we call biosafety law. Then several resolutions on how things move, how we work with this. And finally, which calls my attention to this presentation, is where some people realize, I was not there at that time, it was a gap, I was a few years out, that the new technologies were coming, and we could not put this on a tray and say, okay, let's be there sleeping. We need to deal with that. And so 2015 start working, took 3 years to come up with the normative resolution 16, which is the famous document. So with this, we are now, this is 2018, but we have several examples which I'm going to show you later. So this was also shown. I'm one of these guys here. I think Maria Lucia is one of those here. Well, we are 27 people, all PhD, and we meet every month, 2 days in a month, and we do this. And all are volunteers. This is interesting. So there are 2 chambers, one that deals with human and animal, and one that deals with plant and environment. Every process that goes on this direction goes to a subgroup, more related to the specialty of everyone. Human and animal, they deals with vaccine, livestock, drugs, and plant, crops, trees, microbes, and so on. Funny that in the past, I was there for 2 moments, 2012, I don't know, many years ago, and now again. This was the very discussing group. They were staying the night discussing about Monsanto, Bayer, Dow, Corteva, all these things. Many much discussions. Here was very easy to go and work, not much work. Then I came again, and now this is a very active part of the commission. And this seems to be very quite nice things, technology like GMO for soybean, corn, sugarcane, cotton is more or less the same. It's being more and more well used, and there's no problems anymore. Here, many discussions. So what we do in the daily basis is lab certification, which is a sort of a CQB. A lab has to have the certification to do research, any type of work or manipulation, or even transport, logistics, storing. It has to have the CQB. So the country, the system knows who is dealing with GMOs or related products. We analyze only the projects that are NB2, biosafety level 2 and up. Biosafety level 1 and down, or not down, the biosafety level 1 is analyzed by CIBio, which every institution has its own. So my university has a CIBio. Company Bayer has a CIBio. Every company has a CIBio. So if the thing is classified as NB1, it stays there, only goes up on the annual report. So also we analyze field trials, commercial approval, gene editing consultation, which I'm going to go deep on this presentation, product risk analysis, and there are sometimes technical visits and things like that. So on the second day, we ratify what was discussed in the previous meeting. Everybody has to vote and has to agree on majority. We revise decisions, issue new guidelines, and then give publicity through the official means. So up to now, and this is on the website from the CTN Bio, we have 105, we have GMOs in Brazil are 104 genetic modified plants, being maize, cotton, soybean, sugarcane, eucalyptus, and bean. 56 recombinant vaccines, very important in the livestock sector. 1 genetically modified mosquito, 1 genetically modified salmon, the famous aqua bounty, and 43 genetically modified microorganisms and its derivatives. So in 2015, there was this motivation for analyzing new technologies. This was very smart, I have to say, to see that and spend 3 years in coming up with what we call an RN16 document. So this was involving what we call precision breeding innovation internationally, new breeding technologies, and this was basically framed on our first law from 2005. This is the document. Every time you listen about RN16, is all about this document. So we call TIMP, Technologies of Innovation and Breeding, Precision Innovation and Breeding, which is similar to PBI. It's a new methodology and approach that differs from transgenic genetic engineering by resulting in the absence of DNA and RNA in the final product. And this definition was based, as said before, in the previous law. So it took long discussions to come up with that. So all this document is based in the fact that the previous documents, the previous law states what is GMO and what is not GMO. So it takes into account several biotechnological innovations, novelties, like those listed here. And it's a sort of a consultation letter. If I have an animal which is somehow gene edited, I consult the CTN Bio following a checklist, and I ask if the product is either an animal, plant, seed, semen, embryo, grain, meat, milk, vaccine, doesn't matter, if it's GMO or not. And this is very quickly what is requested. So from the parentals, what is the genetic technology? What is the taxonomic classification, the risk classification, the gene or genetic element, the genetic strategy, molecular characterization, and product expression. So it's all there. You have to answer those questions. And regarding the product, either offspring line or product, the proof of proof of absence of recombinant DNA RNA molecules, which methods you use, how do you assess that? What is the recombinant ability to enter into a target species? So how This system can be used on this species, and if it can go to other species, if it's the case. In the case of animals, is not that much important. The product is commercially approved somewhere, so it takes into consideration, and this has to do with familiarity in a sense, but we don't use that name, only the concept. Oops. If the product used gene drive, it is still something open. There are many discussions about that. And how was evaluated the off-target? It was checked, how you check it, sequence, how much, how deep, and provide that information. So then, company institution send a consultation letter, take 30 days to publicity, decision, it's not GMO, is not a GMO, and it's delivered. Yes or no, there is no maybe. If there's some questions, the CTNBio can go back and please clarify this, clarify that. If it's a GMO, then it follows what was told before, the whole process of evaluation. If it's not GMO, it goes out, and then who is regulating? If it's a medical thing, Ministry of Health. If it's an agricultural product, Ministry of Agriculture, and so on. So some principles behind this process, which was, I would say, new, timely, and very well done, I have to say, because now we are using this instrument. I am using, people are using as a proponent, people are using as a regulator. So it's a prior inquiry on the legal framework. So it's interesting. So you don't continue your research, or you have what we call a legal security. So you're secure to do what you were planning to do as a researcher, as an institution, or a company. So it has very well described parental and product descriptions. Since we are still in the very early phases, we are not inventing new animals or new plants, so it's easy. It describes the techniques employed, so there is no black box. Everything is clear. One thing important, if you have some secret or how do you call confidential parts of this thing, you can say these parts are confidential. So this is removed from the application, and it's manipulated in a separate session, not open to public, and this is the confidentiality is guaranteed. So we need to see how is the construction, where is the modification, how many SNP, how many bases, etc. Precautions with gene drive still. Attention to off-target effects. So this is being more and more enforced. And the analysis is case by case, which has been, since there are few applications yet, is like how it is. Maybe in the future, we'll need to see, well, this was applied, it was approved, now we have another one that is similar or very equal. So just to finalize this, up to now, we have these examples. So some insects that have gene silence, lines of Saccharomyces cerevisiae used in the industrial processes, bull semen with increased muscle, the myostatin, strain of Bacillus thuringiensis, COVID vaccines, new herbicides, tolerance, nematode control, Angus cattle for its leak, sugar cane with changes, I don't remember which one, and tilapia with hypertrophy, muscular hypertrophy, myostatin. So to finalize, I found recently this paper from my colleagues here, some of them, and this is very interesting, very illustrative. So I found it very interesting and very new. And also this document that is from May, it's from May this year, from OECD. There's also some interesting information about, updated information about this. So I hope this is good, and I'm very open for discussions. Thank you.